Okay, I think we'll make a start. Good morning. I'm glad some people got out of bed this morning and made it here on a Friday, so congratulations on achieving that. You've probably, there's notes about these lectures up on Blackboard, so you may have seen them already. But we're going through this morning, continuing on the theme of drugs for oncology and having a look at a couple of old cancer drugs. These are really old, like me, and they are still in use today, like me too. And the whole direction for research in these drugs is to see how we can make them work better, because they have the advantage of being very cheap and they're effective. So first of all, we're going to have a look at thiopurine drugs, and in the process of looking at that, we'll have to revisit the magical world of pharmacogenetics, which hopefully you've come across at some stage, or personalised medicine. And so we'll be looking at that and the problems that occur in pharmacogenetics. So we'll be looking at some of the principles of pharmacogenetics. Firstly, that gene mutations, or they're called alleles if there's different types of mutations present on the one gene, affect how they can affect drug response. And these are usually polymorphic, so they're not rare mutations, they're ones that exist in the population all the time. And that polymorphism can be ethnic specific, for example with alcohol metabolism. We'll have a look at that. We'll see that over the process of both lectures this morning, more than one gene can act on drug response, again using alcohol just as an initial example, although I don't expect you to memorise anything about alcohol metabolism other than how it affects you and how you cope with it. And genes, as a result, can interact with one another in response to a drug. Also, gene exons and introns can modify drug response, as they can modify any gene. So I guess you probably know that from the genetics that you would have done in first year and have revisited along through your course. So not only exons, that's the coding part of a gene, but the introns, that's the so-called junk part of the gene, are now known to affect activity. And at the upper end of genes on the 5' end, you also get promoters, which also modify gene response, so they can modify drug response too. And it's possible to develop a risk score for a drug based on the knowledge of genetic influences. So we're going to start out in what I call pharmacogenetics fairyland, or we could call it, because it was all very lovely in the beginning when we started out in this. And we'll find out what are pharmacogenetics and why did I have a dragon there in the first place. So pharmacogenetics are all about individual drug response, and as you probably know, we all respond to drugs differently. So if you're working in a pharmacy, somebody can come in and say, well, and say, I've got a headache, and you say, well, here's some paracetamol, and they say, but paracetamol doesn't work for me. Well, they're not crazy. There are some people in the population for whom paracetamol doesn't work. Codeine is now a prescription drug because for some people it's perfectly good at relieving pain. For others, it's extremely deadly. And other people don't respond to codeine at all. So pharmacogenetics has also been promises. So we've been promised for years and years now that it's going to change the way we use drugs, and it hasn't so far, just as This says, promise you'll never stop watching anime, and someone says, I promise. There's been a lot of hype about pharmacogenetics as a result in the way genetics has developed in the past 10 years particularly, promising an era of personalised medicine, and we'll look at some of the classic pharmacogenes. You'll know of some of them. TPMT you've probably never heard of, or NUDT. You may have heard of CYP or CYP450. The alcohol drugs, ALDH and ADH, and we'll look at another one you've never heard of called DPYD. But the whole thing that's held pharmacogenetics back is that to be really useful, it has to be fast, because doctors usually want answers straight away. So if somebody's brought into an emergency department in a hospital and they want to administer an antibiotic, they want to know, does this antibiotic produce ototoxicity, which it does in some people, and they want an answer straight away, not tomorrow or the day after. That's becoming possible now through point-of-care instruments. So, as you know, instruments are becoming more and more modified and miniaturized, and it's possible to pack a lot of electronics into things and do DNA tests actually in the clinic. But we all wanted those sort of instant type of genetics that you used to see on the police shows, where they just put a bit of blood into a machine and they can tell you all about the person. So pharmacogenetics is genetics, of course. It's DNA mutations which produce genetic differences, and that produces different phenotypes then. So that's the expression of the genes. So here you can see a lot of different phenotypes, which are quite odd, but nonetheless, we'll see that humans can be just as odd. Pharmacogenes are ones that produce differences in phenotypes of metabolism, transport and receptors. Now, pharmacogenes aren't there to frustrate pharmacists. They're not produced by the body to stop particular drugs working or make them toxic. The genes are there to do other things in the body, and then we introduce drugs into the body that react or rely on those genes. So an example of a metabolic gene, the very first example was discovered in 1889 by this chap with the bushy moustache called Archibald Garrett, and he discovered alkaptonuria, which results, you have missing an enzyme in the body, you accumulate something called homogentisic acid, and as a result your urine turns blue. And that was the first example of a gene that was found to actually produce a metabolic difference, or the first one that people noticed. But in fact, we have lots of them that we see every day. So genetic differences can result in inherited differences in metabolism, and if they're rare, they can result in accumulation or loss of endogenous metabolites, such as alkaptonuria, and it can produce metabolic diseases, they're called, and some can be very serious. But they're rare. But we also carry very common mutations, polymorphisms, and they can also produce differences in our metabolism. And the classic ones are hair and eye colour that we see every day. So we can see here some differences in eye colour and hair colour, and an ability to grow horns. So you can see horns is inherited here in the male line, but not in the female line, it seems. So a polymorphism is a gene that's present in more than 1% of the population. And all the other metabolic diseases are extremely rare, often 1 in a million. Polymorphic genes, like all subject to mutations like all genes, and they can arise from SNPs, single nucleotide polymorphisms, indels. You've probably heard these terms, I'm just reminding you, which are inserts or deletions, and copy number variants. And they can produce differences in drug PK and PD. And as well, let's see, we're going backwards, sorry. The important thing about pharmacogenetic mutations is they remain hidden until you're exposed to a drug. So anyone in this audience here may be carrying polymorphisms which affect their metabolism of a drug, and you won't know about it until you come in contact with that drug, and that's the important aspect of them. And if it's a serious drug, then the results can be very serious. A non-serious drug, well, serious enough is alcohol, and this is a classic one for a pharmacogenetic disorder, we could call it. So alcohol is normally broken down in the liver to acetaldehyde. And then in this diagram, it takes a right-hand turn, and it's broken down first by alcohol dehydrogenase, ADH, and then aldehyde dehydrogenase. And that produces acetate, or acetic acid. And you may remember that acetic acid is just used up in the body in the TCA cycle. So most of the acetaldehyde is degraded, but a little bit seeps through in the bloodstream and gets into the brain, and it's acetaldehyde that actually makes people drunk. It's not alcohol. The acetaldehyde is a very reactive molecule. It condenses with neurotransmitters like dopamine, and it produces neurological effects, and it produces the feeling of being drunk. It also produces vasodilation, and so you get flushing on the cheeks. So you can see the top person here is intolerant, but the bottom one has flushing on the cheeks, and that's due to acetaldehyde. So there are polymorphisms in this pathway, both in alcohol dehydrogenase, ADH, which can be high activity than compared with the normal population, and some people are missing this step here, aldehyde dehydrogenase, and so they don't make acetate. That means that their alcohol virtually all goes down to acetaldehyde, and so from a very small amount of alcohol you produce a large amount of acetaldehyde, and that produces, I wouldn't say being drunk, I don't know what it's like, but I'm told that people feel very sick from it, and they get flushing or vasodilation. So here's a group of tolerant people and a typical response of intolerant people. So this is due to polymorphisms, high alcohol dehydrogenase and or low aldehyde dehydrogenase. I don't expect you to memorize this, but it's just giving you an example so you understand what's happening. So here's an example of alcohol intolerance. It would be, the big problem in this is that there is a polymorphism present in the first place, which we could think of polymorphisms as being dragons, hidden dragons in our population which affect us and we don't know they're there. And it's ethnic specific, so up to 50% of East Asians, particularly Japanese and Koreans and some Chinese groups, have alcohol intolerance because they carry these polymorphisms. They're present in about half the population. It's rare in Caucasians, so it's only about 1 in 100. So amongst Caucasians, somebody who's alcohol intolerant is quite rare, but most people know somebody who can't drink at all. And the people who can't drink, they will just avoid alcohol completely. And I've been in Japan, and you see groups of businessmen all come in and sit down in a cafe, or not a cafe, a restaurant, and they have glasses of whiskey and they all raise their glasses and say "Kampai," but some will just raise the glass to their lips and put it back down again. They won't actually drink. Or some will have, these days, Coca-Cola because they know that it's acceptable not to drink. So it's quite common, but most people amongst Caucasians know somebody who's alcohol intolerant too. And they really can't drink, it makes them horribly sick. Pharmacogenetics, there's also a term pharmacogenomics, which you probably hear about, or genomics in general, and that just refers to a strategy that is often used these days to sequence large numbers of genes, and you can do that for drug response, and it's called a genome-wide association study, or a GWAS, so you've probably heard these terms or come across them, and looking for odd genes that are hard to pin down is a good method, a good way of approaching it using GWAS. And so antidepressant responses, for example, have turned out to be very difficult to track down. The only way to do it is to use a GWAS study. So I suppose the take-home message for alcohol pharmacogenetics and genomics is that more than one gene can be involved in response to a drug. The genes can interact with one another, and as well, they're ethnic specific. So we'll leave alcohol behind, and we're going to move on to our real topic, which is cancer. And I'm going to start out looking at cancer in children, which seems an odd thing, something we don't think about. So if you look at the incidence of cancer by age, along the bottom here, you can see as people get older, the risk of cancer goes up until they get up into their late 80s, and it actually drops slightly. If you get past cancer, then you're more likely to die of heart disease or something like that later on. But if you look right down the bottom end here, there's a little bump from about 1 to 4, 5 to 9. It's high and then it drops back down again. So what's that due? Common, not common, but common enough thing in children are this set of symptoms. And if you're ever working in a pharmacy, in a hospital, for example, and the mother brings in their child and says, oh, they're very short on breath, they can't run around very much, or they're losing weight, they get fevers and they get sweats at night, and they often bruise easily too. Bruising is a result of poor blood clotting, and so it means that your red cells aren't doing their proper job, and that's because you've got too many white cells. So if you have a look at a blood smear on this, normally a blood smear would be full of red blood cells here, but this one's full of these bluish ones, which is just due to a stain, and they're white cells. And they're the symptoms of lymphoblastic leukemia, acute lymphoblastic leukemia, ALL, and very common in children, and then the incidence of it disappears, and by the time you get to about your 20s, you're fairly safe from it until late in age, and then there's a late age form of ALL, which comes on to people my age. So it's an acute, rapidly progressing leukemia. It produces immature lymphocytes in the blood and bone marrow. As a result, you don't have enough red cells. It's the most common cancer in children, peaking at about 2 to 3 years, and it becomes rare by your age, and then it becomes common later on. The drugs, well, children always used to die of ALL. They usually, from the time they were diagnosed, having shortness of breath and bruising and night sweats, they usually survived 3 to 6 months and then they were dead. But the first drug that ever worked to increase their survival was mercaptopurine, which is a thiopurine, which was in the original title of this thing. And it was invented a long time ago in the 1950s. And it's still the most common drug used for maintenance therapy. So we've added other drugs into treating children with ALL. Usually they're treated very aggressively first with chemotherapy. You see them on TV sometimes, the little children, they're sitting in a ward and they don't have any hair because all their hair's fallen out. That's because they're in the stage of being treated with very aggressive chemotherapy to kill off the white cells. And then they go on to mercaptopurine and they have to keep it up for about 2 years, and it's called maintenance therapy. And the survival rate with ALL now is around about 90%. So it's very good. It's one of the success stories in cancer. Soon after, azathioprine was invented. It's a derivative of mercaptopurine, and it was used or invented as an immunosuppressive in about 1960, and it introduced the whole era of organ transplant, and it's now used widely as well for immunosuppression for autoimmunity such as eczema and Crohn's disease. And there's another drug, another thiopurine, which is called thioguanine, and that's been considered dangerous. We'll talk about that later, but it's coming back, so why would we bring a dangerous drug back? It seems weird. So thiopurines have been very cheap. They're out of patent and easy to make. They're effective, but they can be dangerous too. So the question is, how can we optimize their usage? And so thiopurines are a good example of showing us how pharmacogenetics can be used. And early on it was considered there was just one simple gene controlling drug response called TPMT. We're going to discover it's become far more complex, and so that's been the lesson in pharmacogenetics and genetics generally. And they also illustrate how pharmacogenetics can work hand in hand with therapeutic drug monitoring, or TDM. And we're also going to look at thioguanine in the end and how we may be able to bring back an old drug that was considered bad. So, as I said, originally thiopurine fairyland, in this case, was a wonderful place because mercaptopurine was a wonderful cure for acute lymphoblastic leukemia. That's why it was invented in the first place. To understand this, we'll have to look at what a purine is. Mercapto means sulfur, and a purine is what? Do you remember? So I'll revise very briefly here. Purines are the bases in nucleic acids and nucleotides. You know that the DNA code is made up of A, G, C and T, 4 bases, and the A and G are purines, and they look like this. They're called adenine and guanine. Guanine, if you put a sulfur onto it, on the top here where the oxygen was, it's called thioguanine. Unfortunately, the sulfur thing is called mercapto by some people and thio by others, and they've never agreed, and so we're stuck with a stupid system where we have thioguanine and we have mercaptopurine, but it's sulfur in both cases. And the mercaptopurine, it's stuck onto a different purine, which is called hyperxanthine. And these are analogues, then, you know that term too, of normal purine bases. And cells take these up and think that they're normal purines, and they use them by mistake. And they try and incorporate them into their metabolism, and it kills the cells. And so they're called anti-metabolites. And there's a lot of purine analogues besides the mercaptopurine and thiopurine drugs. There's a whole load here: deoxyadenosine, cladribine, fludarabine, and chlorfarabine. These are all anti-cancer drugs as well. And just variations on the ring structures here make these other drugs. But we're going to stick to the thiopurines just for this. I need to revise also how purines are made, and they are synthesised mostly within the cells. They form a nucleotide called IMP, and that diverts into either ATP or GTP. And you've heard of ATP, the energy molecule, and it also the A in DNA and RNA. GTP you probably know less about, but it's in G proteins, and it goes into the G in RNA and DNA. And these are also broken down, so when cells turn over in your body, then the DNA and RNA and the ATP are all broken back down to the purine bases. And in a lot of cells, they're just thrown away, but they can also be salvaged as well. So about at least half of your purine bases are actually salvaged by the body. And thiopurines, mercaptopurine 6-thioguanine, exploit that salvage mechanism. So where the body is normally salvaging purine bases, if you take mercaptopurine, thioguanine, then the cells pick that up by mistake, try and turn it back into nucleotides, in this case GTP, and they make these things called thioguanine nucleotides, TGNs. They have their effect through the G proteins, inhibiting G protein function. And there's also, they're broken down and excluded from the body as cells are turned over. So we'll just make a note here that actually 6-TG is more effective than mercaptopurine, but as we'll see, it's more dangerous. So a typical drug metabolism effect is exemplified by thiopurines, and that is that here's the diagram we've just seen. It all looks lovely, where the drugs go over and make thioguanine nucleotides, which are the active form of the drugs. They inhibit your G proteins, they knock out white cells, and so you get a therapeutic effect. But there's an enzyme called TPMT, and it breaks down about 90% of the drug. This is very common for drugs, as you probably know. Most drugs are attacked by systems in the body which are there to attack xenobiotics. So things that we eat in our diet that the body doesn't like get broken down or removed through urine and so forth. And this is a classic one. It's a methylation enzyme, so it methylates 6-MP and 6-TG. Well, knowing that, that's not a problem really, because it means if you give somebody 100 milligrams of this drug, actually 10 milligrams of it is going to be taken up and used, and the other 90 milligrams is being excluded. This is typical of a lot of drugs that you take. But there's a problem, and that is that TPMT is polymorphic. And so this polymorphism is the dragon that haunts drug usage. So about 11% of Europeans are heterozygous, they're carriers, for TPMT deficiency. So if we have 10 Europeans in the room, probably one of them is a carrier for TPMT deficiency, and that means they have half the activity as normal. That means that they won't respond as normal to mercaptopurine. Not a problem until you come in contact with the drug. So here's TPMT, and it has this polymorphism attached to it. As a result, if it's low, then more of the drug goes this way. So it's the same as we just saw with alcohol. As I explained to you, in most people, alcohol is diverted. It goes to acetaldehyde and then to acetate, and it's used up by the body. But in some people, it's diverted down into acetaldehyde and doesn't get broken down. And in this case, we've got a diversion going on normally, but here, if that's inhibited, more of it is going into the active form. You can sort of draw it this way. So normally, if you have 100 milligrams of 6-mercaptopurine, hopefully you won't have to, but later on in life you might be given 6-mercaptopurine. Then 10 milligrams of it is taken up by your body and the other 90 is broken down or inactivated. So you can draw it like that. Say 10's going this way, 90's going that way. If you're a carrier of TPMT deficiency, then as a rough sort of calculation, you can say instead of 90% going this way, there's only 45% going this way, so only half of it. So instead of 90 milligrams going here, there's 45 milligrams is being methylated or turned into methyl mercaptopurine. So that means the rest of it, 55 milligrams, is going into the active form of the drug. Well, if you look at that, that's 55 milligrams is now becoming activated, whereas normally it's 10 milligrams. That's a massive overdose, and that's the problem that occurs with pharmacogenetics. It's not a small effect, it's a huge effect. And I saw this illustrated when I was working in London in a hospital there for years, and we did TPMT testing. The doctors used to take, gastroenterologists in this case mostly, or there were oncologists and all sorts of doctors, would take blood and send it to us for testing. We'd let them know whether the TPMT was normal or whether it was low, in which case they'd have to adjust the dose. We got blood from a chap, a 50-year-old family man, sent to us. We were so overwhelmed by the number of tests we were asked to do, which were increasing exponentially, that our turnaround time was almost 2 weeks. But that's not normally a problem because most doctors, if you go into a clinic, take blood from you and then say, come back in 2 weeks' time and we'll get the results. So we did the test, found that TPMT was low in this chap, phoned up the doctor to say, well, you know, you can't give him a normal dose of mercaptopurine, and the doctor said, you're too late, he's dead. And he died within 10 days from overdose. So you can see this overdose effect can be very extreme. He developed a leukopenia where it killed off all these white cells, and then you die of infection. But you can get around it by reducing the dose. So you can see here that 55 milligrams is going over into the activation pathway, whereas it should be 10. So if we reduce it about 5 times, then 10 would be going this way. So in that case, instead of giving 100 milligrams, you're reducing that 5 times. 20 milligrams is enough for a patient. So it's often reduced by a fifth or a quarter, something around that. So in laboratories that do this testing, and it's one of the most common pharmacogenetic tests that are done. When I set up the test in London in 1990, in the first year we did 10 patients, the next year we did 100, the year after that we did 1,000, the year after that it was 5,000. They're now doing about 18,000 tests a year and farming it out to other labs. So it's extremely common test, this one. It's not something that isn't used. And it's done by either measuring the activity in the red cell, that's called phenotyping, where you're actually measuring the protein that's produced, or more commonly these days genotyping, so you can measure the mutations of, not measure them, but assess their presence. And there's only 2 major mutations that account for 95% of the patients. But overall there are 31 mutant alleles, and so we recently, a while ago anyway, put them all together and published them so that everyone knows and we also gave them a nomenclature so that we can talk about them. So a general pharmacology question would be, how can we optimise the use of any drug? Not only in pharmacogenetics, we want to minimise its toxicity, improve its response. It's all about dose usually. How do we do that? Do we just guess and say, oh, I think you might be okay on this dose or whatever? That works for a percentage of the population, but not for everyone. Or you can use pharmacogenetics to inform the initial dose and then do therapeutic drug monitoring. It's not done for every drug. It's only done for small numbers of drugs, but it's done for the very dangerous ones. Has it affected the use of thiopurines? The answer is yes. The FDA, Food and Drug Administration in the USA, recommended screening. It's more or less compulsory in the USA, and it's become the same in Australia, England and Europe 12 years ago. So now when somebody's going to be given a thiopurine drug, they're assessed for their genotype or their phenotype. If they're normal, which is about 89% of Europeans, because 11% are abnormal, then you can just normally give them a normal dose, and they check the white cells. That's what they used to do in the past, but now they still do the same, but it assures the physician of a low risk of toxicity. So your patient's unlikely to suddenly die on you, like I saw happen. In 11% of cases, if the patients are carriers, in the past they usually had a high risk of toxicity and could die, and were typically withdrawn from therapy if they survived. Now, on reduced doses, they have a good clinical response. And the problem, if you withdraw a patient from therapy, if they're on one of these drugs, then what are you going to give them instead? If you have a child on mercaptopurine and you're using it to keep their acute lymphoblastic leukemia at bay, and you find that they have a bad reaction to the drug, you can say, oh, well, we'll stop giving you mercaptopurine, but what are you going to give the child? Because if you stop the drug, the leukemia comes back. You've got to keep it going for 2 years. And same with adults. 0 TPMT, heterozygotes are 11%, so 1 in 9. If you remember your genetics from first year, you can work out how many homozygotes are present in the population. So if 1 in 9 in the population, if you're a TPMT carrier, the chances of meeting another one and marrying them is also 1 in 9, so you multiply them together. Then if you have children, because it's a recessive disease, 1 in 4 chance of a child having, one of your children having a complete deficiency, you multiply that all up, it's about 1 in 300 amongst Europeans, so it's quite common. In the past, if they were given a thiopurine drug, they usually died, and certainly you couldn't give them therapy. But now we give them 5% of the normal dose. So it's quite weird where you have a drug that you normally give to a person, give them 100 milligrams and you see it has an effect. But in some of the population, you can give 5 milligrams, a 20th of that dose, and the drug works for them. It's quite strange. So things became better in pharmacogenetics fairyland because TPMT seemed to explain thiopurine variability, and it became a model system in pharmacogenetics. It used to be quoted all the time. However, more dragons appeared, unfortunately, in thiopurine fairyland. A paper came out in 2000 by somebody called Columbell. They looked at a lot of patients and found only 27% of patients with myelosuppression during thiopurine therapy had a mutant allele of TPMT. And the myelosuppression was more often caused by other factors. So suddenly we thought, oh God, you know, TPMT isn't the only answer. So what is? It wasn't so simple. So TPMT definitely caused myelosuppression, but not everyone who had myelosuppression had TPMT, if you can follow that. So it's not the sole cause. So there were unidentified dragons somewhere. What were they? And a clue came out many years later, in recent years, past 5 years. TPMT is almost deficiency, almost absent amongst Asian groups, and yet thiopurine toxicity is high. So we should have picked up on that a long time ago and thought that's a bit weird. Just to remind you, this is what the pathway looks like. Here's the drugs here. They're normally broken down by TPMT, but those that aren't are turned into thioguanonucleotides. We didn't look at this pathway here where they're broken down. And so are there other genes that affect thiopurine toxicity? And what about the catabolic route here? If that's broken, then the TGNs will build up and you'll have more toxicity. And in fact, that's what was happened through one of those GWAS studies in recent years. They discovered a protein called NUDT, which breaks down TGNs, and it's polymorphic in Asia, in Asian patients. This was the original paper that came out 2014, so not long ago, and it was from Korea, and it was looking at a common variant in this thing, NUDT15 it's called, and it confers susceptibility to leukopenia or myelosuppression. Here's another one that came out in 2018 looking at Japanese patients. So this is just happening now. The idea of these couple of lectures, I should say, is to try and fill you in on the latest research. I'm not giving you lectures that have been the same year after year. You're hearing about these things that are happening actually in research at present. And so this is quite new. And it's been found that this mutations in NUDT are strongly associated with leukopenia. So that explained a group amongst, for example, Koreans, very high specificity and sensitivity if you measure NUDT. It affects about half the population. And so NUDT has been found to explain thiopurine toxicity in Asians, which we didn't know before. What else is there? There's another weird protein called MTHFR. These have horribly long names, I'm sorry. And I haven't bothered to give you the full names of them, but because it's easier to learn these just as abbreviations. But this is a protein that controls folic acid metabolism, and MTHFR works here. Then it controls a methyl cycle. Why is that important? The methyl cycle produces a thing called SAMe, S-adenosylmethionine, and that actually provides the methyl group for TPMT. So if you have low MTHFR here, then SAMI becomes low, and then you have low TPMT. So another reason why you can react to thiopurine drugs. Is that a problem? Yes, it is. It shows how one gene affects another, but it's not routinely genotyped at the moment. We're still discovering about this. The problem is the dragon here is that MTHFR is polymorphic and it has a low activity allele in about 60% of the population. Another dragon, a polymorphism dragon that affects our response to thiopurines is called ITPA. And here's our nice diagram again showing how thiopurines work. I don't expect you to remember what these diagrams look like, but just a general idea of these drugs here are either activated or inactivated. The drugs themselves are prodrugs. They're not, mercaptopurine and thioguanine aren't active by themselves. They have to be turned into active things, like a lot of drugs. There's a weird pathway that takes thio-IMP here, turns it into thio-ITP, then breaks it back down again through ITPA, and that's called a futile cycle. It just goes around and around, and why does cells do this? Nobody knows. The answer is cells do weird things. But there's a polymorphic deficiency in 5% of Caucasians and 12% of Japanese, we don't know in Koreans or Chinese at the moment, of ITPA. And if you have that, then you build up this weird thing here called thio-ITP. The other point to this is you notice it's only mercaptopurine and not thioguanine that's affected by this. Maybe thio-ITP is toxic. Well, there was a lot of argument about this early on, but we described ITPA gene mutations. They're both exonic and intronic. You can see there's one here which is an exon 94C2A. This one here, IVS, means intervening sequence. It's an old term for an intron, and both of those affect activity. Either one producing a non-functional protein and the other one changes an alternative splicing branch site and produces low activity. We became interested in this years ago, and there was a lot of argument about ITPA, whether it was actually did anything or not. But now, therapeutic drug monitoring of thioguanine nucleotides has shown that they're very complex. They're methylated by TPMT, and that these things occur. So instead of just a simple metabolite, there's a whole list of them here. There's the TGNs. These ones in blue are the methyl TGNs, so that's produced by TPMT. And you can see over here. But we've found that ITPA interacts with TPMT metabolites, so we get a whole branch of new metabolites appearing, and these have been found recently in patients. This is all recent publication. And so we get methyl TITP. It's awful. So TDM is also, therapeutic drug monitoring is also used routinely to monitor thiopurine therapy. So not only are patients checked first of all for their genetics, but then they're monitored. And so there's been some papers about ITPA, and it's been confirmed it has an effect. This one here is from a very influential group in America. They showed that it affects treatment, produced toxicity in acute lymphoblastic leukemia in children. So The question then is, how do we fight all these dragons and improve the use of thiopurines for lymphoblastic leukemia? And why would we bother in the first place? Well, I think you know why we bother. First of all, the drugs are used widely, not only for ALL, but they're also used for autoimmune diseases like inflammatory bowel disease, IBD, for Behçet's disease, for eczema. Behçet's is a neurological disease. They're also used as immunosuppressants. They're cheap and effective. The alternatives for these is all the new monoclonal drugs and biologicals. They're very expensive. But what about thioguanine? So to finish up, we're just going to quickly consider thioguanine, because that was the sort of Cinderella of the thiopurine drugs. It was the one that was always left behind. It was considered the nasty one. Initially, when thioguanine was tried out, this is back in 1993, for acute lymphoblastic leukemia. This is a very influential group in England. They said, in the context used, 6-TG, that's thioguanine, appears well tolerated, produces higher concentrations of the cytotoxic metabolites than 6-MP. And for children who are resistant to the traditional drug, it may be the preferable alternative. That's taken from the summary. So that was good. So with thioguanine, things looked happy in thiopurine fairyland, but then a nasty dragon appeared, and that was liver toxicity, and it was started to be called a dangerous drug. So this group produced thioguanine, a paper, this is in 2003, 3 years later. It causes serious liver injury in inflammatory bowel disease patients. Another paper from an American group, early hepatic nodular hyperplasia, that's liver disease, associated with 6-thioguanine therapy. In 2006 to 10, so quite recently, TPMT, this is thiopurine methyltransferase, that's what TPMT is. Genetic polymorphism is associated with thioguanine-related veno-occlusive disease. That's another term for the liver disease that's produced. What happens is the liver gets blocked up, a veno-occlusive disease, or it's called hyperplasia as well. And that was in children with ALL. Another group, toxicity and efficacy of thioguanine versus mercaptopurine. They found at the bottom here, 95 patients developed veno-occlusive disease of the liver, and 82 of these were assigned to thioguanine, so the majority of thioguanine patients had veno-occlusive disease, and they said that it was nasty, caused excess toxicity. Finally, this influential group in America in 2010 put the nail in the coffin, saying toxicities of thioguanine preclude its protracted use. So basically at that stage people said you shouldn't give it anymore. So it was abandoned. However, thioguanine was actually rescued in 2013 by research done here at UQ, surprisingly. Here's a paper that came out then. A novel mouse model of veno-occlusive disease was developed. This is a group here. We did the study by giving thioguanine to mice and then looking at their livers. And along the bottom here, there's controls, thioguanine, mercaptopurine at different doses. And so these are a measure of damage to liver. So this is sinusoidal damage. this is central vein damage, and basically you can see 6-TG or thioguanine damages the liver. So the mice got very ill and died being given thioguanine. So that confirmed thioguanine was nasty, and we were giving 2.5 milligrams per day to the mice, whereas on the other hand with mercaptopurine they were fine on 5 milligrams. But we found by splitting the dose, so giving 1.25 milligrams or a half dose twice daily, BD, suddenly all that toxicity disappeared and the levels of liver disease dropped right down. So that was a very simple answer to this, why this drug was causing problems. Just by splitting the dose, the liver wasn't exposed to a very high concentration of the drug in one go. Simple. I don't know why no one thought of it before, but we did. So here's a critical assessment. This was in 2017, just last year, an important paper in Journal of Digestive Diseases, looking at treatment of inflammatory bowel disease, but it can be used for leukemia as well. Is it time to rehabilitate? Thioguanine treatment. It looked, amongst other things, at other studies that people published in the past and found they gave these high doses, 40 to 80 milligrams of thioguanine, produced a lot of toxicity here, but when you lowered the dose to half, then the number of patients who experienced toxicity dropped off rapidly. So that was published, splitting a therapeutic dose, a simple thing. Two-centre study was done between here in Pace and also in London. And in particular, this was used for bowel disease, ulcerative colitis, which is very resistant to treatment by all the modern drugs, suddenly was very responsive to thioguanine. So this was a marvellous thing. This is Crohn's disease, which is also an inflammatory bowel disease. So that was a combination from London and Brisbane. And splitting the dose, so we've just got patients to actually break the tablet in half. It's not very high technology, but in the Netherlands, the Dutch have gone ahead and produced simple half doses and quarter dose tablets. They call it Thio 6. And here you can see they're looking very happy about it. This is a rather happy dragon. This is actually from a Dutch slide. Thio 6, the Dutch say, produced a major breakthrough in the formulation in Netherlands, used for inflammatory bowel disease, but now starting to use it for leukemia as well. And they produce it in 10 or 20 milligram tablets. It costs about €1,000 a year for a patient, but for patients taking monoclonals, it costs them 10 to 15,000 euros a year. So far it's saving the Netherlands between 15 and 30 million euros a year on monoclonals. And the Dutch have started publishing this stuff, so there's one paper by them with lots of Dutch names on it, saying that it's very safe. Then another one